The structure of the operon in prokaryotes include a regulatory gene, promoter, operator, protein repressor, RNA polymerase, and the genes. The regulatory gene produces the protein repressor. The protein repressor is responsible for binding to the operator whenever necessary. The RNA polymerase is responsible for coding the genes and making proteins. It does this by scanning the DNA strand. The promoter is the spot where the RNA polymerase binds to the strand of DNA. The operator is where the repressor binds to it whenever it needs to stop the RNA polymerase from going forward. An inducible operon has a regulatory gene, repressor protein, promoter gene, operator region, and inducers. Lactose is an example of an inducer. In an inducible operon, the operon is not binded with the repressor at first. The repressor could be removed when the inducer binds to a repressor and changes its shape. The role of the repressor is to block the RNA polymerase and stop it from encoding a series of genes behind the repressor. This is how we are able to not show some of the traits that our genes are supposed to have, to encrypt us to have. The ability to decide whether a gene should be turned on or off is a very important trait. Complex multicellular organisms, such as our cells, need to have. As we all know, all of our cells contain the same DNA. It is the ability to turn on or off certain parts of the gene strand that allows for each type of gene to have specific traits that are related to its function. For example, cells in the stomach contain genes that tell us to produce HCl. The ability to selectively activate certain regions of the gene is also the reason why muscle cells look different from eye cells.